Thanks for the invitation to speak today. I understand I have a little bit more time, so I may dwell on some of the slides a little bit longer than I would otherwise. I hope that's okay. What I want to talk today about is emerging diseases in bumblebees, but this area of research sits within my sort of broader interests. And so largely I'm interested in how complex host parasite systems work. So a lot of the work that has been done in host parasite systems looks at single host species and single parasite species to try to understand how they interact. And I'm more interested in what happens when you have a single host with multiple different parasites, all of which are trying to survive and infect that host, and what happens when you have multiple host species dealing with a single parasite, and what happens when you bring it all together, when you're looking at multiple host species and multiple parasites, all of them interacting in these complex epidemiological dance. And the work that I'll talk about today is a small aspect of that, but I'm very happy to answer questions about bumblebee parasites in general at the end. Okay, so emerging diseases in general, I think, are one of the major conservation threats at the moment. And so there are lots of examples of these. So white nose disease in bats, which is an emerging fungal disease, which is wiping out bats in North America, is a classic example. And emerging diseases affect just not wild animals, but they also affect humans as well. So Ebola is another example of an emerging disease. And so emerging diseases, understanding their dynamics, understanding why they happen and how they happen, is a really big question in ecology and also in evolution, with real ramifications for human health. And the reason that they're interesting in bees is because bees are pollinators. And so there are obviously a whole array of organisms that pollinate plants, ranging from insects like bees and flies and beetles, through to birds, through mammals, reptiles even. But the vast majority of pollination and the vast majority of pollination service is provided by insects and predominantly provided by bees. And when we're talking about bees as pollinators, we have to distinguish between different groups. And so I think most people, when they think about bees, think of honeybees, hopefully not as ecologists, but most people outside of ecology tend to think that all bees are honeybees. And honeybees are obviously highly social. They're also, in North America and in Europe, essentially domestic or managed animals. There are no or very few wild colonies left, and I'll talk about why in a minute. Or in the northern hemisphere, a lot of that pollination is being provided by bumblebees, which are again a social species, but annual social species. So unlike honeybees, which are perennial, bumblebees have annual colony cycles. And then the final group of bee pollinators are the solitary bees. And just to put this into context, there are about, depending upon who you ask, depending upon the taxonomist, around 10 species of honeybee in the world. There are about 250 species of bumblebee, and there are about 19,500 named species of solitary bee. So that perhaps puts some context around when we talk about bees as pollinators. And they're important in natural ecosystems, obviously. They provide pollination services to flowering plants. And for many of those flowering plants, those pollinating services are essential for them to set seed. But they're also important from a human health perspective. And so there have been a variety of attempts to measure this. This is one particular attempt by Gallig and colleagues. And they suggested that globally, pollination services are worth about €153 million per annum in terms of the crops that they're helping to produce. Now, any calculation like this is actually wrong, and it's wrong for a number of reasons. One reason it's wrong is because we don't actually understand who is providing pollination services to many crops and the importance of self-pollination versus insect pollination versus wind pollination. And the other reason is that it's wrong is because obviously as the cost of growing crops changes, then the cost of them in shops changes, and therefore what people are prepared to purchase changes. So any estimate of value is always going to be inherently wrong. But what we can say is that there is some value, and that value is probably quite large. And so it matters to us that we have healthy pollination services, which, as I said, are predominantly provided by bees. And therefore, it should concern us from that perspective that a lot of pollinators are in decline. And this is not a new story. It's something I'm sure you're all aware of, but just to emphasize it, those declines have been measured both qualitatively and quantitatively across a range of bees. And so we know that honeybees are in decline, not globally. At a global scale, managed honeybee colony numbers are going up. But in North America and in Europe, so in those temperate regions, managed honeybee colony numbers are going down. And there are a range of reasons for that, which I'll talk about. In terms of wild honeybee colonies, as I said, in North America and Europe, they're essentially gone, so we can view that as a complete collapse. Whereas if you go to South America or Africa or Asia, there doesn't seem to be a major problem there in terms of honeybee numbers. So very often you hear in the media about global honeybee colony collapse, and that's actually a lie. What we have is regional colony declines, and in other parts of the world, colony numbers are actually going up, as far as we can tell. And the data those sorts of claims are based on comes from things like the Food and Agricultural Organization numbers, but also data from local beekeeping groups as well. Bumblebees have also been suggested to be in decline, and we have good data in terms of the range of species. So we have no good quantitative data on abundance for bumblebees at all. So we know that the ranges of some species have declined, and you can go out to sites where there used to be, say, in the UK, 24 species, and you go out there today, 30 years later, and there are only 6 species left. So we know that ranges are collapsing. The best data for this initially came from Britain, from just mainland Britain, and that was largely because in the 1800s there were lots of priests who didn't have very much to do with their time, and so they would go out and they would collect insects and pin them to describe the natural world, which I suppose was vaguely religiously motivated because they were trying to describe the wonder of God's creation or something like that. And so we have that huge database, which means that now we can go out and look at bees and we can say, well, okay, these bees used to be here and they're not anymore. Those data were backed up by data from a number of other European countries, and more recently there have been some fantastic data produced in the United States and Canada and also in South America, which are also showing collapses in ranges of bumblebees. We have no idea about abundance at all, but we do know that ranges are collapsing. In terms of solitary bees, we know almost nothing, and that's because people don't collect data on them, and they're very small, and they're very difficult to identify normally. We know that for 2 very small regions, one in the Netherlands and one in England, that in those 2 areas, solitary bee numbers have collapsed, but we don't have a broader picture of it. And so it's very hard to say what's really going on with solitary bees, but probably they're also going through declines. And we know what the reason for the vast majority of that, well, we know what the historical reason for that decline is. And that decline is driven by things like habitat loss. So here is a classic bee pollinator habitat. It's a flowering meadow. This one happens to be in Britain, but it could have been anywhere throughout Europe. This is the kind of habitat that bees rely on. There's lots of resources. Those resources are available throughout the year. There will be nesting sites nearby and sites for them to hibernate. And we've seen huge losses in these habitats, which has been largely driven by industrialization of agriculture. And so we see these very florally diverse, very structured, complex structured habitats being replaced by large-scale agriculture, and that's happening at a global scale. Yep. So it happened first in North America and Western Europe, but it's now happening in Africa. It had happened thousands of years ago in Asia. So we're seeing habitat loss, and that's clearly driving a lot of these declines. If you take away the food and you take away the habitat, then the animals can't exist anymore. But what I'm interested about is the aspects of those declines that can't be explained by this habitat loss. And this is where emerging infectious diseases become important. And I'll talk first about what is known in honeybees. So this is the head of a honeybee pupa a few days before it emerges. And this is the mite Varroa destructor. And this is a classic story of an emerging parasite or an invasive parasite. So for those of you who don't know, Varroa destructor is originally a parasite of the Asian honeybee, Apis cerana. And in Apis cerana, it's co-evolved for millions of years, and it has a fairly low impact on its host. Apis mellifera, the western honeybee, was moved over to Asia because its colonies are bigger, it's a better pollinator. And when it was moved over in the early 1900s, this mite saw its opportunity and shifted across. So this is fairly typical of most emerging parasites and diseases. There's an anthropogenic cause at the very beginning. We moved a species to where it shouldn't be, and then a parasite jumped across to it. It turns out that in Apis mellifera, the western honeybee, this mite actually has a huge impact, and that's because of the colony dynamics. So the mite essentially reproduces in the cells of the honeybees, and it does that growing off the larvae that are in there. So it's a bloodsucker. It bites into the larvae in the pupae and sucks out blood. And in the Asian honeybee, the only cells which are large enough for the mite to do that are the ones that males are reared in, because males are bigger than females in these species, and those cells that they grow in, those classic honeybee cells, are larger. Males are only produced at one time in the season, and so in the original host, in Apis cerana, the mite has a very short period of time to go through reproduction, and so its population can't climb too high. The European honeybee, Apis mellifera, is a bigger bee, and therefore the cells are larger, and therefore the mite can reproduce in the cells of worker honeybees. And workers are produced all year round. And therefore the mite population explodes, and basically, if the mite population explodes, it wipes out the colony. And it does that partly because of sucking blood out of larvae, and so weakening the bees. But the other reason that it does it is because it acts as a transmission route for viruses. So this is a honeybee that has deformed wing virus, and you can see why it's got that name. Its wings are basically completely crippled. It can't fly. It's useless. It's no good to the colony. It will also live for less long. It will die much more quickly. And that's because it has deformed wing virus. Now, deformed wing virus, along with many other honeybee viruses, has always been present in honeybees, but its original transmission route would have been through the bees ingesting viral particles. And what the mite does is it comes along and it sits on either a larva or an adult bee, and when it bites to suck blood, it transmits the virus. Just like a malarial mosquito, when it bites blood in a human, transmits malaria. And that's a new transmission route for the virus. It gets into the hemolymph, so into the blood of the bee. It can't be controlled there, and it explodes. And that's what causes this huge damage. And so essentially what we've got here is a double emerging parasite disease system. Firstly, the mite emerging, and then the mite's emergence causing the emergence of the viruses. And this is what is driving the vast majority of honeybee loss in North America and Europe, and it's what's wiped out all of the wild populations of honeybees. And we can be pretty confident about saying that. And so we've known about this for a long time. As I said, the mite crossed over in the early 1900s. It spread globally. We've got a very good track of how it spread globally, and it spread globally through honeybee keeping practices. So when people moved hives or when people sent queens around. So we know how that spread around the globe, and we know how that impacted honeybee populations. More recently, it's been suggested that emerging diseases may also be playing a role in the elimination of other wild bee populations. And two examples of that come from North America and also from South America. We know from pretty good data that ranges of a suite of North American bumblebees, so not all of them, but a subset of them, have hugely contracted over the past 20 to 25 years. Much more rapidly than European bumblebees have declined, and apparently not linked to habitat loss, because all the habitat loss seems to have happened a long time before the bees declined, in very broad brushstrokes. And we also know more recently that this beautiful animal here, this is Bombus dalbomii. It's the largest bumblebee in the world. It's about that kind of size. People often refer to it as a flying mouse because it's this lovely brown color and it's really quite large. This is a native of South America, and the range of that species has declined hugely, and it's quite likely that it will go extinct in the next possibly 2 or 3 years. So it's collapsed from ranging all the way through Chile to a tiny area essentially in Tierra del Fuego. And those declines have been linked with parasites. So the North American declines have been linked with the potential emergence of a fungal disease called Nosema bombi. And in South America, they've been linked with the potential emergence of a trypanosome called Cratidia bombi and a neogregarine intracellular parasite called Apocystus bombi. And in both of these cases, it's been suggested that these are emerging diseases. So in North America, the story goes something like this. Commercial bumblebee rearing, which was used to produce bumblebees for commercial pollination of crops like tomatoes, squashes, and soft fruits, collapsed in the 1980s, collapsed due to a fungal infection, this fungus here. And the idea that's been suggested is that that fungus got out from those commercial bees into the wild bees and spread as an epidemic across North America and essentially knocked down the populations of a subset of North American species that were susceptible to it. The data that provide support to that are correlational. So surveys done by Sydney Cameron and her colleagues showed that if you look now, or in the last few years, at species which have declined, they have a very high prevalence of the fungus, and the species that haven't declined have a very low prevalence. But that doesn't actually tell you anything about cause and effect. It could be that species that have declined are weak for another reason, and therefore they have higher levels of the parasite. So there's some correlational evidence there, but no definitive data that any kind of emergence has taken place. And the same thing is true in South America. At the same time as you get the collapse of the bee population, you get an increase in the prevalence of these parasites, and they possibly have come from, again, commercial bees which have been naturalized in Chile. But again, it's really fairly loose correlational data. But both stories do suggest that this kind of emergence may be happening. And in particular, interestingly, in South America, this neogregarine parasite has been suggested to be spilling over not from bumblebees, but from honeybees. So a cross-genus transmission route. And that, combined with a couple of other papers which suggested that parasites and diseases that have always traditionally been thought of as honeybee parasites might be spreading into other pollinators, led us to ask whether there were emerging infectious diseases coming from honeybees to bumblebees. So could it be that honeybees are not just pollinators, but also a source for disease for wild bees? And so to do that, we did a study across mainland Great Britain. So we've got 26 sampling sites here. The majority of them are on the mainland, from the south up through the north. There should have been a few more sites up here, but the year in which we did this study, the weather was absolutely rubbish. I know you probably think it's always rubbish in Britain, but honestly, the sun does occasionally shine, particularly down in the south. This year, it basically rained and was cold for the majority of the year in Scotland, so all of our sites up here, there wasn't a single bee present, so we couldn't do any sampling at all. The only two non-mainland sites, from here and from here, are sites where the honeybees don't have this Varroa destructor mite, and therefore they don't have high levels of viruses either. And that'll become important in a bit. At each of these sites, we went out and we sampled and we collected bumblebees of 2 species, the 2 most abundant species, and we also collected honeybees. And you don't need to worry about the colors and the letters here. They simply represent the species that were collected. And I'm going to collapse those data anyway, so you don't need to worry about them. I guess the important point is we weren't just looking at a single bumblebee species, we were looking at the assemblage that's actually present. And what we looked for was two potential emerging diseases. The first of which is a fungal disease, Nosema ceranae, which again is an emerging disease in honeybees. It's come from Apis cerana, the Asian honeybee again, come into Apis mellifera, and has emerged in European populations and got up to quite high levels in the last 10, 15 years or so. And there was a record of it being present in bumblebees. So we thought we'd look at that one. And we also looked at deformed wing virus, again, because it's emerged in honeybees over the last 15 or 20 years because of the Varroa mite, and because, again, there were the occasional record of it being present in bumblebees. And so we collected all these bees, we brought them back to the lab, we screened them all using molecular tools to see what the prevalence was, and then we looked to see if we could understand the patterns of prevalence. And to do that, we used models where we used the prevalence in honeybees as a potential predictor of the prevalence of these parasites in bumblebees. And our argument for doing that essentially is that we know that deformed wing virus is present in very high levels in honeybees, and we know that Nosema ceranae has become present in very high levels. So if there's any directional transmission, it's presumably coming from where it's very abundant to where it's less abundant. And these are the first set of data. So these are the data for Nosema ceranae. These are predicted prevalences, so predicted from the data that we collected. And the darker colors indicate where the prevalence of the particular parasite is higher. So this is Nosema ceranae in honeybees. And the darker color shows where you would expect the prevalence to be higher. So here in the southwest of Wales, but also in areas in East Anglia. And this is Nosema ceranae in bumblebees, and here the prediction is that the prevalence would be much higher in this whole East Anglian area. And the models suggest that when this fungus is present at relatively low levels in honeybees, it predicts quite well its prevalence in bumblebees. But as soon as that prevalence in honeybees gets higher, it seems to swamp any pattern out. And that could be because the spores are simply present at such a level in the environment that there's no pattern at that point. So once the spores reach a certain prevalence in the environment for the bees to pick them up from the flowers on which they're foraging, any pattern disappears. So there's a weak relationship here, but not a terrifically strong one. I would feel insecure saying to you that Nosema ceranae is emerging in bumblebees from honeybees. However, when we turn to deformed wing virus, we see a very different and a much stronger pattern. So again, this is its prevalence or predicted prevalence in honeybees. And you can see that there's really strong levels down in southwest England, across the southern part of Wales perhaps, and again in East Anglia. East Anglia seems to be a really bad place if you're a bee. There's lots of parasites there. Not really sure why that should be. It's probably due to local climatic conditions and also the structure of the landscape. There's a lot of mass cropping going on in these areas. This is its presence in bumblebees, and you can see that there's a correlational match here. This is the presence when it's actually replicating. And so what other studies before have done is simply looked at the presence and absence of the virus, and that doesn't actually tell you whether it's an infection or not. It just tells you whether it's there. And if it's there, it could be being vectored, because these parasites are picked up on flowers when the bees are foraging, and it could be it's just picked up the virus on its surface, or it's drunk it, and then it's going to defecate it later on and not get infected, or it's just going to move it from flower to flower. And so because these are single-stranded RNA viruses, you can actually look to see if it's an active infection by looking for the opposite strand, so the replicating strand of the virus. And that's what we did here. And you can see that this pattern of replication matches really well the prevalence in Apis. And when you do these statistical models, there's a really strong predictive power. So if you know where honeybees have deformed wing virus, you can be really sure that it's going to be present in bumblebees at the same site. The other interesting thing to notice about these graphs, which hopefully you've seen, is that the prevalence levels in honeybees go up to 80 to 100%, and that's what we would expect because we know that deformed wing virus is very abundant in honeybees because of this mite, and its prevalence levels in bumblebees are much lower, so going up to maybe 25 or 15 to 20%. And that again backs up the idea that the parasite is flowing from the honeybees to the bumblebees. It's going from high prevalence into a population where the prevalence would naturally be much lower. However, this doesn't tell us necessarily that it really is that pattern. It could be that the bumblebees are just more resistant and that in some areas transmission is easier because of the structure of the landscape again. So if you've got areas where there's just a single crop and all of your pollinators are foraging on it, the chance of transmission is going to be much higher than if there's a very diverse array of flowers where bees can actually partition where they forage and split those resources. And so to see whether it really is direct transmission from honeybees to bumblebees, we did some sequencing and created a phylogeography for this virus, and that's what this is here. The large colour blocks here represent sites. So this is one site, the purple is another site, this browny colour is a third, and the yellow is a fourth. Inside, the sequences of the viruses taken from bumblebees are shown in blue and from honeybees in black. And if there is transmission going on at these sites, you would expect the bumblebee and honeybee viral clusters to cluster together within a site, and that's exactly what we see. So here in this site, the bumblebees and the honeybees share exactly the same viral sequences. We see exactly the same thing here in the purple. We see the same thing happening in the brown site. We see it in the brown site again here with a different variant of the virus. So there's clearly transmission going on at those sites, and that transmission seems to be being driven by the honeybees, pushing those viruses into bumblebees as the bumblebees are foraging on flowers. So that's fairly good evidence for an emerging disease, but what impact is it having? If the virus doesn't actually impact the bumblebees, we don't really care. It's changing the epidemiology, and that's interesting for me as a scientist, but it doesn't have any real applied interest. And so we conducted laboratory experiments, and a number of other people have done this too. And the first thing, obviously, that you want to show is that the virus and the fungus can actually infect bumblebees. And so we took, so our controls are here. The green is the fungus, the black is the virus, and this is showing prevalence. And these are our treatments where we inoculated the animals by collecting virus particles or fungal particles from infected honeybees and feeding them directly to the bumblebees. And you can see that the prevalence goes up both for the fungus and for the virus. So yes, you can infect the bees, but also the virus has a negative impact on bumblebees. So this is a survivorship analysis. The blue animals are ones that were inoculated with the virus. The red ones are ones that are not. And you can see that there's a significant decrease here in the longevity of bees. Bumblebees actively forage for about 21 days. And the difference that we're seeing here in longevity, the impact of the virus, would have a significant effect in the field on the fitness of colonies if the bees are only living, well, they're living about 40% less long. So that's about half their foraging life gone. So we think it's likely that these viruses, which are emerging, are also probably having a significant impact on the bumblebees. And then we took this further and we looked across an array of honeybee viruses, and we basically see the same pattern across all of them. So these are rather complicated graphs, but I'll just go over them really quickly. These are data for the same sites for a range of different viruses. So here's DWV that we looked at before, but there's also black queen cell virus, acute bee paralysis virus, slow bee virus, and sac brood virus and slow bee paralysis virus. And this is the data when it's all clumped together. But again, we're seeing this strong correlation between prevalence in honeybees and prevalence in bumblebees, with generally, for most of them, the prevalence being higher in honeybees than it is in bumblebees, although not all, interestingly. So this leaves us, I think, with a suite of questions that we haven't answered yet, but that we hope to answer in the future. The first is, what really is the directionality? So the models that I showed you made the assumption that it's most likely to come from honeybees into bumblebees because it's higher prevalence there. But really, we'd like to nail that down. We really want to know, is it really that direction? And so what we plan to do in a study that will be starting next year is to actually go out, sample again for not just bumblebees and honeybees, but a broader array of pollinators, screen them for the viruses, but then do very deep sequencing on those viruses. And because we know what the mutational rate of the virus is, we can then construct real-time evolutionary epidemiology models where we can really say the virus is coming from species A into species B. And that's really important because if we can nail down that directionality and it is coming from honeybees, we can then give management advice to honeybee keepers as to where to put their hives to minimize the impact that they might be having on our wild pollinators. Whoops. What happened? The second thing we really want to know is we want to be able to extrapolate those impacts on individuals. So this is a bumblebee with deformed wings. I'm actually cheating a little bit. This one doesn't have deformed wing virus. It has deformed wings because of a different infection. But it gives the right impression. We want to know whether those individual impacts are having population level impacts. So can we extrapolate from the impact of a virus on an individual bee to the survival of a colony, but then to the population biology of the organism? How many colonies, how many individuals have to be impacted before we see an effect on the population dynamics of wild bees? And we also would like to know, is this actually a much broader phenomenon? So we've shown this relationship in bumblebees and honeybees, but there are reports out there of some of these viruses being present in solitary bees and also in hoverflies, in syrphids, all of which are important pollinators. And so what we'd really like to know is actually, are we dealing with a much larger potential meltdown due to emerging disease in pollinators as a group, or is it restricted perhaps just to these social bees? And again, we're hoping to get at that in a project which is starting fairly shortly. And I'd like to end just by saying thank you to all of the people who did the work with me, particularly Matthias First, who was a postdoc in my group, who did the vast majority of the study I just talked about, and our funders, obviously, and to you both for the invitation to come and talk to you and also for your attention. Thanks very much.